Hello everyone, welcome to my channel Dr. Tapati's presentation. Today topic of the presentation is industrial production of alpha-amylase. Already I have uploaded 2 videos related to alpha-amylase. You should watch those videos. Alpha-amylase is one of the most popular and important enzymes with wide industrial applications. This enzyme is endo-amylase because it acts on internal alpha-1,4 glycosidic linkages present in starch molecules, producing low molecular weight products of oligosaccharides with varying length with an alpha configuration and alpha limit dextrins. This type of enzyme is mostly extracellular in nature. There are mainly 5 major steps involved in alpha-amylase production. First of all, selection of suitable alpha-amylase producing microorganism either from bacteria or fungi. Second step is inoculum preparation. Third step, production of alpha-amylase in solid-state fermentation process or submerged fermentation process. Fourth step is purification of alpha-amylase using various downstream processing methods. And finally, applications of alpha-amylase in various sectors including food industry, detergent, paper, textile, biofuel and pharmaceutical industries. In this video, I am going to discuss about mainly selection of suitable alpha-amylase producing microorganism, inoculum preparation and production of alpha-amylase in SSF and submerged fermentation process. Few important criteria for the selection of microorganism are: microorganisms should produce maximum alpha-amylase activity within short incubation period. Microorganisms should grow on cheap substrate. Microorganisms should not be pathogenic in nature. That means it should be safe to handle. Alpha-amylase should be extracellular in nature for more product recovery. And minimal amount of other metabolites should be produced during the fermentation process along with alpha-amylase. Alpha-amylases are universally distributed throughout the plant, animal and microbial kingdoms. Animal sources of alpha-amylases are primarily used for physiological functions within the body to help digest dietary starch, while microbial sources are more widely used for commercial production and industrial applications. A number of microbial sources exist for the efficient production of this enzyme, but only a few selected strains of fungi and bacteria meet the criteria of commercial production. Bacillus species is widely used for thermostable alpha-amylase production to meet the industrial needs. A number of reports are available on alpha-amylase production by different fungi, but the most thoroughly studied and industrially useful source is Aspergillus species. In spite of the wide distribution of amylases, microbial sources are used for the industrial production due to various reasons. Those are: microbial sources are easily available and growth rate is faster. They are generally cheaper to produce. Their enzyme contents are more predictable and controllable. Reliable supplies of raw materials of constant composition can more easily be arranged. Plant and animal tissues contain more potentially harmful materials than microbes, including phenolic compounds, endogenous enzyme inhibitors, and proteases. Genetic changes using recombinant DNA technology can easily be done on microbial cells for elevated enzyme production and scientific development. So these are the different advantages available from the microbial sources. So industrially, microbial sources are used for the alpha-amylase production. Among all the fungal sources of alpha-amylase, Aspergillus oryzae is the most important and popular species and has been used as yellow cozy in producing many traditional fermented beverages and foods such as Japanese steak, miso, and soya sauce. The two major fermentation industries, Novozymes and Danishko Genencor, all employ Aspergillus oryzae as their primary production organism. Taka-amylase A or TAA That is the major enzyme produced by this mold, and it is famous worldwide to be a leading enzyme for industrial utilization and academic study. Three main processes are involved for the selection of suitable alpha-amylase producing microorganism, either bacteria or fungi. Those are isolation, screenings, and identification. Starch-rich residues are the better potential source where alpha-amylase positive microorganisms can be isolated. That means those microorganisms could be isolated from places like cassava farms, soil and processing floor factories, starch processing wastewater, bakery industrial area, etc. There are several methods of isolating pure culture of bacteria, including streak plate method, pure plate method, spread plate method, and serial dilution. Techniques used for isolation of fungi from soil include serial dilution agar plate, workup soil plate, syringe inoculation, immersion tube method, screened immersion plates, plate profile, hyphal isolation, soil washing, partial pre-sterilization, soil sieving, flotation, bedding, etc. Isolated pure cultures are screened for alpha-amylase activity by flooding the starch agar plates with iodine solution. This shows clear zone around amylase producing microorganisms. The zones of clearances are measured as an indication of the degree of amylolytic activity. The diameters of the clear zones over the diameters of the colonies are measured using a ruler as the halo is to colony ratio. Higher halo is to colony ratio indicates greater production. So higher halo is to colony ratio producing microorganisms they are generally selected for the production of alpha amylase and identification of that pure culture. Once a pure culture is up, in various tests and analyses can be performed to identify the specific microorganism. This includes biochemical tests like tryptophan breakdown test, microscopic examination, and genetic analysis. Let's know about various steps involved in inoculum preparation. During the first stage of inoculum development, the inoculum is taken from the working stock pure culture to initiate the growth in a suitable liquid medium. That means primary culture inoculum preparation. In this process, suspension of vegetative cells, spores, hyphae is prepared in sterile tap water or sterile saline. Then secondary culture inoculum is prepared from the primary culture in small conical flasks containing liquid media. Inoculum development is done in sequential step to increase the volume of the initial inoculum to the desired level. That means finally seed fermenter is used for the inoculum preparation in a large volume. Fermenter capacity is increased according to the requirement of inoculum volume for the enzyme production process in industrial scale. So whatever inoculum is prepared in the fermenter, it is utilized for the production of enzyme either in solid state fermentation condition or in liquid fermentation condition. Fermentation is the chemical transformation of organic substances into complex compounds by the action of enzymes produced by microorganisms like yeast, fungi, or bacteria. Commercial production of amylases is carried out in various steps, especially because the environmental factors required for the optimum growth of the microorganism may differ from those required for the production of the enzyme. Although specific fermentation procedures adopted by manufacturers vary to a certain degree, but there are mainly two principal methods. Submerged fermentation method, in that case liquid media is used, and second method is solid state fermentation method where solid substrate is used. Both the processes are used for the commercial production of alpha amylase. Let's know about differences between SSF and SMA processes. In case of SMA process, the substrate is made soluble or suspended as fine particles in a large volume of water. That is, in SMA process, liquid media is produced, whereas in case of SSF process, it is performed on a non-soluble material that acts both as physical support and source of nutrient in the absence of free-flowing liquid. In case of SMA, substrates are used like soluble sugars, molasses, liquid media, fruit and vegetable juices, sewage, wastewater, synthetic media, etc. But in case of SSF, solid substrates are used like wheat and rice bran, wheat and rice straw. That means mostly different agro residues are used. Cassava, barley, oil cakes, sawdust, bagasse, paper pulp, fruit and vegetable waste, coconut coir, etc. Products are secreted directly into the production broth. In case of SMA process, additional solvent is not required to extract the product. Whereas in case of SSA, the microorganisms involved synthesize enzymes that degrade the polymeric substances into smaller and more easily digestible compounds. Extracellular enzymes and other products are secreted into the solid fermented substrate. Products are extracted by adding solvent in the fermented solid substrate, or water can be added in the fermented solid substrate to extract the enzyme. In case of SMA, it is suitable for microorganisms like bacteria, which need more moisture content for their growth. Whereas if you consider about the SSF, filamentous fungi are commonly used due to their relatively high tolerance to low water activities and osmotic pressure conditions, their high potential to secrete hydrolysate enzymes, and their morphology. In case of SMF, it is more suitable for the extraction of secondary metabolites which are required in liquid state. Whereas if you consider about the SSA, various bioactive compounds are produced which are used in food, pharmaceutical, cosmetic, fuel and textile industries. This table is showing different microorganisms used in SMF and SSF condition for the alpha-amylase production. As I told you, in SMA, mostly bacteria is suitable microorganism. So in alpha-amylase production also, bacteria is suitable organism for SMA process, and mostly Bacillus species is used. In that case, examples are Bacillus amyloliquefaciens, Licheniformis subtilis, Stereothermophilus, and Bacillus brevis. Whereas if you consider about the fungi, mostly Aspergillus species were used for the alpha-amylase production in SMA. Those are Aspergillus oryzae, Niger, and Aspergillus flavus. Other than Aspergillus species, other fungi are also were also used for the production of alpha-amylase production in SMA process. Whereas if you consider about the SSA process, in that case, mostly Aspergillus species is used, like Aspergillus oryzae, Fumigatus, Tamari, Aspergillus flavus, Aspergillus starius, and Aspergillus avamori. Other microorganisms were also used for the production of alpha-amylase in SSF condition. Examples of bacteria used for the alpha-amylase production in SSA are Bacillus amyloliquefaciens, Licheniformis, subtilis, Polymixa, Mesenteriosus, Bacillus bulgarus, Bacillus coagulans, and Bacillus megateria. So these are the different examples of microorganisms used in SMF condition and SSF condition for the alpha-amylase production. There are several advantages in both the cases, that means in SMF as well as in SSF processes. If you consider about the advantages of SMF, those are easy medium sterilization and end product purification, easy to control the process parameters like medium pH, temperature, moisture, oxygen transfer or aeration, agitation. SMF is most suitable method for the use of genetically modified organisms. Fermentation may be carried out as batch and continuous mode in SMF, and in this process lesser space requirement is there. Whereas if you consider about the different advantages of SSF, those are simple and cost effective, less water demand is there, high concentration product can be recovered from the SSF process. Nutrient rich solid materials can be recycled because most of the cases waste products are utilized for the fermentation process. That means mainly agro residues are utilized, those are very cost effective and those can be recycled. This process resembles natural habitats of fungi and bacteria. That means it mimics the natural environment of the microorganisms. Less release of effluent, so it reduces pollution. Lower sterility demand, lower catabolite depression, lower energy demand for heating. Mixed culture possible for synergism of metabolic performance in case of SSF process. So in last few decades, various types of SSA products, including food and food ingredients, agro-industrial products, pharmaceutical products, and many more have been documented. If you consider about the alpha-amylase production, both the processes are fruitful in industrial scale. So you can observe a lot of experiments on alpha-amylase production in both the cases, in SMF as well as in SSF processes. Although solid-state fermentation process is relatively rare in Western countries, it has extensively been employed since ancient times in the Oriental, Asian, and African countries for the production of fermented foods, started inocula, mushrooms, dough fermentation, etc. Several research groups found that SSF is more advantageous for alpha-amylase production as compared to SMF. Aspergillus oryzae (SMC), Aspergillus kawachi IFO 4308, Pycnoporus sanguineus, rhizobus species, Aspergillus oryzae, Aspergillus starius, NCFT 4269. These are the organisms which showed better result in SSF as compared to SMF for the production of alpha-amylase. This table is showing alpha-amylase production in SSF by various fungi. According to best of my knowledge, Aspergillus oryzae IFO 30103 was the best alpha-amylase producing strain among all the fungi, producing 12,899 U per GDS alpha-amylase activity in wheat bran during the solid-state fermentation process. Whereas this table is showing alpha-amylase production in SSF condition by various bacteria. According to this table, Bacillus species PS7 is the best alpha-amylase producing bacteria with 4,64,000 U per gram dry bacterial bran alpha-amylase production in wheat bran substrate during the solid-state fermentation process. During actual production of alpha-amylase in SSF or SMF, first step is substrate selection, then optimization of fermentation condition by one factor at a time and by statistical optimization method. Substrate selection plays an important role in production of alpha-amylase in SSF as well as in SMF. In case of SMF, soluble substrates are used. Those are soluble starch or sugars, molasses, fruit and vegetable juices, sewage wastewater, and synthetic media. Sometimes insoluble substrates are also used in SMF, but it is used in very little amount. Whereas in case of solid state substrate fermentation process, wheat and rice bran, wheat and rice straw, cassava, barley, oil cake, sawdust, bagasse, fruit and vegetable waste, brewer's paint grain, etc. are used as the main solid substrate for the production of alpha-amylase. Pre-treatment of solid substrates has often been found useful in improving its digestibility and access for microbial attack in case of solid state fermentation process. Most of the cases it is observed that wheat bran is the best substrate for alpha-amylase production as well as other enzymes and organic acid production as well and other biotechnological applications because of few reasons like high initial starch content of wheat bran, very good buffering capacity, good water holding capacity and better aeration, low cost of wheat bran and its composition assortment. After substrate selection, optimization of fermentation condition by one factor at a time is carried out. Then statistical optimization method is followed. Various physicochemical parameters have been reported to affect the production of alpha-amylase by SMF and SSF. Among these, the most important and thoroughly investigated factors are the temperature, pH of the medium, incubation period, inoculum age, aeration, moisture content and bed height of the substrate for solid-state fermentation process, agitation for SMF, phosphate concentration, surfactants, metal ions, and additional carbon and nitrogen source. In my next video, I will discuss in detail about influence of physicochemical parameters on alpha-amylase production. This is an overview of alpha-amylase production process by solid-state fermentation or a summer fermentation process. First of all, selection of suitable alpha-amylase producing microorganism, either bacteria or fungi. In that case, isolation, screening, and identification, these are the major steps involved. Inoculum preparation means primary culture inoculum, secondary culture inoculum, and then seed fermenter. After that, fermentation process is carried out either in SSF condition or SMF condition. So this is the principal method of production process, where substrate selection and pretreatment is the first step. Then media preparation and process parameters optimization by one factor at a time, and statistical optimization method is carried out. Purification method means in that case various techniques are used like centrifugation, ultrafiltration, precipitation, chromatographic techniques, and electrophoresis methods. After purification, characterization of the enzyme done by checking the effects of concentration of substrates, concentration of enzyme, temperature, pH, concentration of products, time, activators, cofactors, coenzymes, surfactants, inhibitors, light and radiation. After characterization, product packaging and storage, and finally application of the enzyme in different industrial sectors like food, detergent, paper, textile, biofuel, and pharmaceutical industries. Upstream processing means from selection of suitable microorganism to actual fermentation process. That means production of alpha-amylase in SSF and SMF condition. That is the so this up to this step upstream processing. And downstream processing means from purification to application step. Okay, so total upstream and upstream processing as well as downstream processing together, these two processes are involved for the alpha-amylase production. Thank you very much for watching this video. Kindly like, share and subscribe this channel.